In this video, we are going to talk about the regulatory function of the T cells. So the acquired immunity consists of two arms. One is the humoral immunity, which includes the production of antibodies, and second is the cell-mediated arm of the immune system. In the antibody production, T cells are regulating the function of the B cells. The antibody production can be divided into two different events. First is the T cell dependent response, and second is the T cell independent response. First, we will talk about the T cell dependent response. In the T cell dependent response, protein antigens are presented to the T cells, the helper T cells. First, the APCs, which include in this case the B cells, those B cells will engulf the bacteria. They will fragment those bacteria and their antigens, and then they will present their antigens on their surface. And now those antigens are presented to the helper T cells. The helper T cells will recognize those protein antigens, and they will produce interleukin 2, interleukin 4, interleukin 5, and interleukin 4, which is a B cell growth factor, and interleukin 5, which is a B cell differentiation factor, will further grow the B cells and allow them to differentiate into the plasma cells, which will further produce antibodies. So the interaction between the B cell and T cell is further made stronger by different proteins, which include CD28 molecules on the surface of the T cells, which will interact with the B7 molecule on the surface of the T cell. Furthermore, LFA1 on the T cell will interact with the ICAM1 on the B cells, and there are also ICAM proteins on the T cell that will interact with the LFA protein on the B cells. So as a result of T cell dependent response, all classes of antibodies are produced, which include IgG, IgM, IgA, IgD, and IgE. Now let's talk about the T cell independent response. So the helper T cells can only recognize protein antigens. They cannot recognize the polysaccharide antigens. So in case of polysaccharide antigens, those antigens are directly recognized by the B cells through their B cell receptor IgM and IgD, and the B cell will prepare or make antibodies against those polysaccharides on their own. And in the case of T cell independent response, primarily IgM is produced by the B cells. So next is a cell mediated immunity. In the cell mediated immunity, the antigen presenting cells will present the antigen to the helper T cells. Helper T cells will then produce interleukin 2, and interleukin 2 is further involved in the activation of other lymphocytes, which include the helper T cells and the cytotoxic T cells. So this was the regulatory function of the T cell in the cell mediated immunity. There is an other function of the T cells, which is also categorized under a regulatory function of the T cells, which is the suppression of the certain immune responses. So there are certain T cells that can suppress antibody production, and failure of such regulation may result in unrestrained antibody production to self antigens, and this can lead to autoimmune diseases. There is no specific population of T cells that is mediating and suppressing the immune response, but there is evidence that in some situations CD8 cells can suppress the immune response. There is also a balance between the CD4 and the CD8 cells, and if this balance is disturbed, it will lead to diseases such as, for example, AIDS. In acquired immunodeficiency syndrome, the normal ratio of CD4 to CD8 is more than 1.5, and this ratio is greatly reduced in the case of AIDS. Many CD4 cells are destroyed by the human immunodeficiency virus HIV, and the number of CD8 cells increases, and this imbalance, which is the loss of the helper activity and increase in the suppressor activity, results in a susceptibility to opportunistic infections and certain tumors. The MHC class 1 and class 2 is also playing a very important role in the immune response, but there are some viruses that can affect the amount of MHC in the body, which includes the hepatitis B virus, adenovirus, and cytomegalovirus. They can prevent an increase in the class 1 MHC protein expression, thereby reducing the cytotoxic T cell response against cells infected by these viruses.